Hello, I'm Arthur Georges, and I'm going to talk today on sex reversal as a driver of rapid transitions between modes of sex determination. But before I start, let me thank the conference organisers, Djivan and Michail, for inviting me to speak today. Now, I think we're all aware that reptiles have an astonishing diversity in modes of sex determination. We have species with genetic sex determination, that is sex chromosomes, with both male and female heterogameity. We also have an array of species with temperature-dependent sex determination, or TSD, whereby the sex of the offspring is determined by the environmental conditions within the nest. And we also have some parthenogens, both facultative and obligatory. So reptiles are a very fertile area for research. Most of what we know about genetic sex determination comes from studies of therian mammals. Mammals have a dominant sex gene, SRY, which resides in the Y chromosome, and when it's expressed, it causes a runaway upregulation of another gene called SOX9, which drives the regulatory processes leading to the formation of the testes, and also cross-suppresses the female pathway. Now, in mammals, the sex chromosomes are heteromorphic in the sense that the Y chromosome has degenerated, and this has been evolved with other mechanisms such as dosage compensation and execution of genes, which lock in the GSD. The Y becomes indispensable. Now, in birds, we have a dosage system involving a gene called DMRT1. It's present on the Z chromosome, and in double dose, it generates a male phenotype, but in single dose in the ZW individuals, it generates a female phenotype. Now, DMRT1 is like SRY in the sense that it promotes the runaway upregulation of SOX9 and the promotion of the male regulatory pathway leading to a testes, and again, it cross-suppresses the female pathway. And sex chromosomes in most birds are heteromorphic. GSD in reptiles is a work in progress. Basically, we have no sex-determining gene yet identified. There are some candidates, but the sex-determining gene probably differs across species. There's also no runaway upregulation of SOX9 in males. So what we found in our transcriptomic studies is that SOX9 is upregulated in both males and females to a point, and then it remains high in the males and declines substantially in the females. This is dramatically different from what occurs in mammals and birds, and so we can't necessarily translate our learnings from mammals and birds to reptiles. And in particular, some of the genes at the headwaters of sexual differentiation are likely to be quite different in reptiles than they are in mammals and birds. Many reptiles have homomorphic sex chromosomes, unlike birds and mammals. And as I've said, some of the expectations we have from mammals and birds are unlikely to apply to reptiles. Now, TSD in reptiles is of particular interest because there is no overt genetic underpinning to sex, and sex determination is fundamentally an epigenetic, not a genetic process. Transitions between TSD and GSD are relatively common, and we know this because TSD and GSD are haphazardly distributed across the reptile phylogeny. Even closely related lineages can have one with TSD and one with GSD. Now, we used to think that TSD and GSD were mutually exclusive mechanisms, quite different in their function. But now we've come to understand, I think, that TSD and GSD reside at the endpoints of a continuum of states with intermediate forms having gene-environment interactions determining sex. Now, a question arises as to whether those intermediate forms are transient or whether they're evolutionarily stable. So we've been working on the dragon lizard, the central bearded dragon, Pogonovidiceps, and it has genetic sex determination with female heterogameity. So when you incubate the eggs at between 22 and 32 degrees, you get a 50-50 sex ratio, which is what you'd expect for a GSD species. However, when you push the temperatures higher, you get sex reversal. And it's sex reversal of the ZZ genotype to a female phenotype. And the higher you push the temperatures, the greater the frequency of sex reversal, until at 36 degrees, you've basically got 100% sex reversal of ZZ individuals. Now, these ZZ individuals that have been reversed, they're females and they're fertile. So not just viable, they're fertile. And so when you cross a ZZ female with a ZZ male, you obviously get all ZZ offspring. And if they're incubated at 28 degrees, you get all male offspring. However, if you push the temperature up, say to 34 degrees, then sex reversal locks in and some of the ZZ offspring become female. So you get mixed sexes at 34 degrees. And if you incubate them at 36 degrees, all of the ZZ individuals are reversed to female. So basically what we've done in one generation in the laboratory is we've converted a GSD species, a ZZZW species, to a TSD species where temperature alone determines the sex. This is quite remarkable. Now, the sex reversal is not just a laboratory phenomenon. We've looked across a range of the species and found multiple sites where sex reversal has occurred in nature. And so the process of this transition from GSD to TSD could well occur in nature. Now, one of the really interesting things about this is that overproduction of females by sex reversal can drive down the frequency of the W chromosome by invoking Fisher's frequency-dependent selection. So we had a look at this in Pagona, and we used the equations of Bull 1981 and validated them for Pagona, and then generated this plot. So at temperatures between 30 and 32, basically the equilibrium state for the frequency of the ZW genotype is 50-50, as you'd expect. But as you increase the temperatures, what happens is that the frequency of sex reversal increases, and that invokes frequency-dependent selection to drive down the frequency of the ZW genotype. And eventually you lose the W chromosome altogether. And interestingly, our study site at Cunnamulla has Pagona right on the precipice of that transition between GSD and TSD. Now, it's important to note that this trajectory here is a trajectory of the equilibrium state, which means that at a particular point on this trajectory, it's evolutionarily stable. It can persist indefinitely. And that's been reinforced by the work of Lisa Schwanz, who did some more sophisticated modelling in the Journal of Evolutionary Biology last year. Now, we get a similar situation with Bassiana duperrii. Now, it's an XXXY system, and sex reversal of the XX genotype occurs at low temperatures. So it's a mirror image of Pagona, but the same process can be modelled. And basically, as temperatures decline, you get a precipitous drop in the frequency of the XY genotype, and ultimately can get the loss of the Y chromosome altogether. Now, at our study site at Mount Giannini, again, our study site, the animals are on the precipice of losing the Y, but there's also another site which we haven't yet sampled, which is much higher, Mount Gingera. And at Mount Gingera, the temperatures are such that we would expect the loss of the Y chromosome. So basically, this loss of the W or Y chromosome is something that can occur under the conditions that are experienced by these animals in nature. So if we have a look at this question, are these intermediate states transient or evolutionarily stable? Well, not necessarily. They're not necessarily transient because both of our approaches to modelling suggest that intermediate states can be stable and persist indefinitely, including cryptic retention of sex chromosomes. And I point to the papers by Sarah Whiteley on the Jackie dragon which provided evidence of cryptic sex chromosomes in that TSD species, and the work of Jodie Wiggins on the collared lizard, who similarly provide evidence of cryptic sex chromosomes in a TSD species. Now, as another question, is TSD an evolutionary sink? Well, perhaps not if cryptic sex chromosomes lurk, waiting to reinstate should opportunity present itself. Or if residual machinery predisposes particular chromosomes to readopt a sex determination role. And I point to the work in chromosome research by Dennis O'Malley that looked at that question. Another question is GSD an evolutionary sink, as one would expect looking at mammals and birds, but perhaps not if many reptiles can jettison the Y of the W chromosome and still produce the formerly heterogametic sex. And again, Sarah Whiteley in PLOS Genetics this year has mapped out the alternate regulatory pathways that determine sex after the loss of the W chromosome. So I refer to that. Another question, is conventional selection necessary to drive transitions? Well, no, transitions can follow a neutral pathway under Fisher's frequency-dependent selection alone, acting on sex reversal. There is no need to invoke specific adaptionist explanations to transitions. Focus, at least in Begona, turns to what selective forces consolidate and maintain the new state once the transition has occurred. So in conclusion, I just point out again the remarkable evolutionary lability that occurs in reptiles and their sex determination, which provides unparalleled opportunity for research, particularly in the species where gene-environment interactions are involved in determining sex. And I'll leave it there with acknowledgement to my team on the right there, and thank you for listening.
Today I will present one of my works from my PhD project, which is about effects of sex-limited experimental evolution, and in this case on the transcriptome. So the main aim of the PhD project is to observe the initial steps of XY, and in this case male, and then ZW and female sex chromosome evolution from a hermaphroditic ancestor. And in nature, usually separate sexes evolve by acquiring sterility mutations, and the most common is a recessive male sterility followed by dominant female sterility and leading to the XY system, and vice versa is for the ZW system. And here is the classical model of the sex chromosome evolution. We are mainly interested in the very beginning, as it's usually hard to study the initial steps of sex chromosome evolution because most sex chromosomes are at least several million years old. So we have applied an artificial like GFP as a sex determining gene, and then we want to observe if it resembles like a new sex determining gene arousing on a new sex chromosome, and then does recombination change and gene expression and other things are happening next to the region. So, and then the XY systems usually evolve faster than the ZW systems based on the fact that there is usually stronger selection on males. So besides the sex chromosome evolution, it follows with sex-biased gene expression, but also separate sexes can evolve gradually by investing more in one sex role and decreasing investment in the other sex role. So now we specifically looked at the gene expression, and we used Macrostomum lignano as a model organism to study these initial steps of heterogamety. And our main prediction is that there should be reduced recombination around the sex determining GFP locus, and that the genes located nearby should change in expression and so on. So Macrostomum lignano is a small 1.5 millimeter long sea worm occurring in Mediterranean It is obligatory outcrossing, simultaneously hermaphroditic and reciprocally mating. And it has 4 chromosomes, where the large chromosome, where we think the GFP insert is located, is actually a duplication of all 3 small chromosomes. So it's a hidden tetraploid, and its genome actually can be variable. And here you can see that the worm has many sex-specific organs, like male organs in blue and female organs in pink, and I will come back to that later. So our setup involves 3 treatments: the male-selected treatment, which is the pseudo-Y, the female-selected treatment, which is the pseudo-W, and then we have the control treatment, which is an equal mixture between both. And then we have 4 replicate populations of each containing 48 individuals. And what we do, we mate the focal worm, which contains this pseudo-determining GFP gene. And then we mate it with 2 wild-type worms. And in the male selected lines, we pick out the white worms, and the green progeny they produce, which we keep for the next generation, have gotten the GFP through sperm. So it resembles the Y chromosome, and the vice versa for the female selected lines, where we take out the green worm for egg laying, and the green juveniles are kept, and they got the GFP through egg cells, which resembles the W chromosome. So how did the transcriptome respond to this treatment? So after more than 20 generations, we did full seq of 40 individuals per population. Also, we did RNA seq on our ancestral GFP lines on Illumina technology. And then for bioinformatics, we did the quality control with Nextflow, then trimmed adapters with Trim-O-Matic. Then we decided to assemble our own transcriptome based on these ancestral GFP lines, because the reference genome and transcriptome is based on highly inbred line, which contains extra large chromosomes. So we wanted the alignment to be more specific, and as it's our ancestral line, so it should even increase the specificity more. So it was done with Trinity and the alignment with Salmon. And then we looked for differential gene expression using HR. Then we looked for functional annotations for GO terms, KEGG pathways, and sex organ specific or biased genes from previous studies. And then we wanted to look specifically on the GFP gene expression and the genes located on the same scaffold as the GFP locus. So predictions are pretty straightforward, that female biased genes should be higher expressed in female selected lines and the least expressed in male selected lines, and vice versa for male biased genes. And here you can see the results, that the largest difference in expression is between female and control lines, which is a bit surprising, knowing that usually males evolve kind of faster, but as our experimental evolution setup contains only mating group of 3, and in regular lab conditions and in nature, these worms mate often and with many more partners. So the sperm competition was very limited, and that means that our male selected lines could actually be more feminized than the stock populations or the natural populations. And you can see that some transcripts have changed in expression quite a lot. So they are supposed to be biologically relevant. And specifically, it's interesting to look at these 234 transcripts where they should be differentially expressed between females and both controls and males. And these 84 should be then truly male specific, where they should be expressed differentially, higher, for example, in males and lower in females and controls. And then we found one transcript which followed all the predictions that it was higher expressed in female selected lines, the least expressed in male selected lines, and it was known to be ovary biased. But in humans, it's also like sex biased gene. However, in humans, it's like it is the transmembrane protein 72, and it's kidney specific and more male biased with expression in testes. So it could mean that this specific gene is participating a lot in like sexual cascades, but during animal evolution can diverge either in male or female direction. Then when it comes to functional annotations, we found that metabolic changes have changed the most, especially for proteins like amino acid synthesis, then also carbohydrates like carbon metabolism. Then despite the fact that female selected lines changed the most, the actual change was mainly in the testis specific genes. So these female selected lines decreased the expression of testis specific genes. And then there is an opposite pattern for ovary specific genes, and maybe females have higher immunity than controls based on these Go terms. So here you can see expression of sex organs specific transcripts. And here, for example, 13 transcripts are highly expressed in male selected lines compared to females, which are known to be testis biased, here in the figure. And 2 transcripts were higher expressed in females than in males, which are ovary biased, but we did not see any other change in other sex organ specific transcripts. And then I did also PCA to see if it can differentiate the lines. And for testis biased genes, you can see there is differentiation, that females tend to be on the left side and males on the right, and controls in the middle, and the same for the ovary biased genes. And for the GFP, we did not find that it has changed in expression, which is good. That means that we haven't really selected on higher GFP expression itself. However, we also did not find any change in gene expression which are located next to the GFP, so on the same scaffold. So as the main conclusions, we can conclude that female selected worms have responded the most, and it could be because we had low level of sperm competition. Then our RNA sequencing results have showed that the largest changes are in metabolic pathways and testis function, and that maybe we have uncovered many more sex role bias genes in this species, and that we can use this selection protocol to sexually differentiate this hermaphroditic worm quite successfully. Yeah, then I want to thank you for listening to my colleagues and collaborators, and yeah, if you have any questions.
So amniotes is an excellent model for studies considering sex chromosome evolution. Among others, amniotes demonstrate a notable variability on sex determination systems, because there are lineages with environmental sex determination and other lineages with either male or female heterogamety. Although the construction of the ancestral sex determination system is still under debate, what is probably so far well known is that there are more than 20 different GSD systems across amniote lineages. During the last years, we tried to explore the evolution of sex chromosome in amniotes using a diversity of methods. So we work with cytogenetic methods in order to identify the presence of GSD in previously unstudied lineages. Then we applied DNA-seq, RNA-seq, or other NGS methodologies in order to identify the gene content of the sex chromosomes in these lineages. And in a third step, we usually apply a qPCR test for homology. qPCR can be used to compare the gene copy difference between males and females. And for the X or Z specific genes, the homogametic sex is expected to have double number of gene copies in its genome in comparison to the homogametic, to the heterogametic one. During the last years, we tried to gather information and answer a number of questions. Among them was, what is the level of stability of the GSD systems in amniotes? For example, it was well known for many years that mammals and birds seems to have nicely differentiated sex chromosomes which were stable for long term. However, there were contradictory opinions about the rest of the amniotic lineage, especially on squamate reptiles. Then we tried to explore if the age or the degree of sex chromosome differentiation can be connected to stability. If there is any systematic difference between male and female heterogameity on the stability of a GSD system, and at the end, if the evolution of sex chromosomes can be random or not. Consider the stability of the GSD system. We work on several mainly reptilian lineages, and I will not go through all our last projects, but the most recent ones. So recently we documented, for example, by qPCR that gopodid geckos share a homologous XXXY sex transmission system, which is more than 46 million years old. A few years ago, we used the same qPCR test across representative species from phylogenetic scale of snakes, and we find out that sex chromosomes of canophidian snakes are conserved for more than 76 million years. Same methodology was also applied in aguimorphan lizards, and the result was that varanids, helodermats, and alligators share homologous sex chromosomes. Nevertheless, there was an exception. There was an inner lineage. At that time, it was published only Agui fragilis, but now we know that there are some more anguids around this phylogenetically related to Anguis fragilis that seem not to share the same sex determination system. Similar image we find also when we studied iguanas. So anoles and the majority of the iguanas have XXXY system, which is quite old, almost 100 million years, with Gallus 15 chromosome. This enteric block corresponds to the X-specific region. In the next of the presentation, I will discuss about homology between different amniote lineages using as reference the synteny to chicken chromosomes. This is typical for studies in amniotes because chicken was the first sapropsid genome that was sequenced, and up to now probably the best annotated. And furthermore, the chicken chromosomes seems to reflect well the ancestral synthetic blocks. So anoles and the other iguanas had homologous sex chromosomes stable for a long time, and then there was a second lineage, the basilisk, which had also XXY. However, the X-specific region is homologous to Gallus 17. Here in this phylogenetic tree, the basilisk are being depicted as an inner clade. Nevertheless, the phylogeny of iguanas is a little bit controversial, it's not fully resolved. There are phylogenies that also depict these two groups as sister clades. Overall, not to spend more time going one by one the lineages, what's important is that there are almost 11,000 non-avian reptiles, and only 5% of them seems to have ESD. 60% of reptiles belong to one of the 5 lineages with stable sex chromosomes. So the majority of the species diversity is either skinks, which have XXY system 85 million years old, canophidian snakes, iguanas, lassids, or monitors. Now the rest of 40% of reptiles, there are of course few small lineages that are still unexplored. There are many lineages with with limited number of species, which we know that they have some stability. And of course, there are a few more lineages which show variability. What comes after we know that there is stability at least to the lineages that were old and have nicely differentiated sex chromosomes? The question is if the age and degree of sex chromosome differentiation might play a role in the stability. So we explore another lineage, skinks, which the result was quite interesting because skinks are cytogenetically poorly differentiated, the sex chromosomes, and they have homomorphic sex chromosome. You can see here from the cytogenetic data that CGH, cooperative hybridization between males and females in these two species, Skincus skincus and Tropidophorus baconi, you can see that it's not visible any clear sex-specific region. The only difference that might be visible between the X and the Y in Skincus skincus is a polymorphism in the copy number of rDNA loci. So what we do is we sequence the genome of a male and a female Skincus skincus, and we calculate the male to female gene coverage, which revealed 37 Z-specific genes, all of them homologous to Gallus Gallus 1. A qPCR test of homology across phylogenetically informative species of skinks showed that all major phylogenetic coverage, the majority of the phylogenetic coverage of skinks has homologous XXSY system, which can be dated 85 million years. We're just missing to test now a few more species, which is a more basal lineage, but this is a forthcoming project. These results are similar to previous results published more or less in the same period from Cornello Paramo et al. In 2020. So from skinks and some other lineages, probably we can extract the conclusion that even poorly differentiated sex chromosome can be stable in long term. From reptiles, we have also lineages where they are relatively young. However, they have nicely differentiated sex chromosomes, cytogenetically visible, and those seems to remain stable at least for that small period of time that have evolved. Recently, we work in such lineages as the carphodactylid geckos. So carphodactylids are have cytogenetically differentiated heteromorphic sex chromosomes. They have a ZZW system. We explore two groups. One is the Antigundissaurus and Nephrurus clade. These two genera have chromosomes homologous to chicken 10 and 17, and this group appear to be approximately up to 30 million years old. Then there is a sister clade, Sartorius, which has chromosomes homologous to chicken 17 and 24, and this system seems to be approximately 20 million years old. We don't know if these two clades, they have homologous sex terminal system. They share a common region in their sex chromosome, this Gallus Gallus 17. Nevertheless, the age of these systems should vary something between 15 and 46 million years. After collecting all the information from bibliography, including putting together our data and of course data from other people working on amniotes, we can now extract conclusions for more or less 27 lineages where the gene content of sex chromosome is known. Here you can see a phylogenetic tree with all these lineages and also the age of each of these GSD system. So we try to get some impression if the type of heterogamety might be involved on stability, and probably there is not such a pattern. It seems that both XXY and ZW systems can be stable in long term. What was our most probably interesting question after the previous three was if the evolution of sex chromosome is random or not. So what we did is, you can see here this figure. We project for each of these 27 lineages of amniotes with known sex chromosome gene content. We group their chromosomes according to synthetic blocks. We define 35 synthetic blocks based on chicken chromosomes. As I said before, we use chicken as a reference because many of its chromosomes seem to represent the ancestral state. So these are 33 chromosomes minus the chicken chromosome 29, which is probably poorly annotated, and so far in the assembly there is not protein content, plus the linkage group F, which is from an early genome project. It's a genomic region, which is sex chromosome in boas. However, this region is not assembled well in chicken genome. And also we split the chicken chromosome 4 to two synthetic blocks, 4P and 4Q, because the chicken chromosome 4 is a homomorphy of chicken. It originates from a recent fusion of two chromosomes. So the main output of this figure is that we realize that there are synthetic blocks that appear multiple times as sex chromosomes, to be part of the sex chromosomes in different lineages. For example, Gallus 17 appears to be part of sex chromosomes in at least 5 lineages, Gallus Z in 4 lineages, Gallus 4P in another 4 lineages, and then chromosomes 1, 2, and 5 appear to be part of sex chromosome in 3 lineages each one. What is probably interesting is that at least in Gallus 17, 4P and Z, there are genes like NR5A1 is the gene that before Arthur George spoke about, DMRT1, SOX3 or adenosine receptor, which are genes that are well known to play the role of sex-determining locus in different vertebrates. So probably these synthetic blocks, they have some advantage to develop a sex chromosome because of hosting genes that can affect the gonad development. Of course, the problem is that there is no study so far in reptiles where it have been identified accurately the sex-determining locus, so this is only speculation. What's also important from this table is that there are synthetic blocks like the chicken chromosome 3 or chromosome 8 and 9, which never contribute to sex chromosomes. So what we did is that we try a multinomial test using air package exnomial to test the goodness of fit of the observed distribution with the expected frequencies. So what we did then is that we tested 3 hypotheses. One of it was if each synthetic block has equal probability to emerge as chromosome, as sex chromosome. If each synthetic block has probability to become sex chromosome depending on the number of genes that it's hosting, or if the synthetic block has probability to become sex chromosome depending on its physical size. The result was that all 3 hypotheses were rejected, and we interpret as that probably some chromosomes are more likely to become sex chromosomes. This is it's not a new idea. It was discussed also in the past in bibliography. There is a very nice review from Jenny and Katie from 2010, and it seems probably that the selection of synthetic blocks, the emergence of synthetic blocks to sex chromosome in amniotes probably is not random. So reaching the conclusions, in the question if the GSD systems can be stable in amniotes, especially outside mammals and birds, so the answer is probably yes. Of course, there are some exceptions where we have some rare turnovers from one GSD to another. The stability of self-determination system doesn't seem to be correlated much to the age or the degree of sex chromosome differentiation. We didn't see any obvious pattern of differences on stability between male and female heterogamety, and the evolution of sex chromosome seems to be non-random. Before finishing my presentation, I would like to thank all my collaborators in this project, and also you for your attention, and I'm available for questions.
My name is Robert Baird, and I'm a PhD student at the University of Edinburgh. And I'm going to talk about the focus of my PhD project, which is on the evolution of probably one of the weirdest sex determination systems out there, which is that of the fungus gnat. So fungus gnats are a family of flies with a host of really unusual genetic properties, including the presence of germline restricted chromosomes, DNA puff formations in the salivary gland polytene chromosomes, as well as elimination of the entire paternal genome such that males don't pass on their genes to offspring. But they also harbour one of the most peculiar sex determination systems where females control the sex of offspring. And in the species Brydesia coprophila, males are exo, but females are monogenic, meaning that they produce offspring of only one sex. So you have androgenic females who produce only sons, and gynogenic females who produce only daughters. And a series of cytological experiments in the 1970s revealed that the factor that distinguishes the two types of females is a long, non-recombining inversion-based supergene that extends most of the length of one X chromosome in gynogenic females, denoted the X prime chromosome. And the chromosome cycle of this species is fairly complex and needs a little more elaboration. So whereas in females, meiosis is normal, in males it's irregular and results in the production of diploid sperm through X non-disjunction. And as a result, all females have the same chromosome constitution. They have 3 X chromosomes. And then whether a zygote develops into male or female depends on how many chromosomes are eliminated from the soma during early embryogenesis. So this occurs around the 7th cleavage. And zygotes that lose 1 X chromosome develop into females, whereas those that lose 2 X chromosomes develop into males. And the X prime chromosome is inherited in a sort of regular Mendelian fashion, such that the gynogenic females, the female producers, produce both gynogenic and androgenic females in equal amounts. So the aims of my PhD have been to sequence and identify the X prime inversion, identify a master switch that controls sex determination through chromosome elimination, and to understand the evolutionary causes and consequences of the system in a sort of broader context of understanding the evolution of sex determination mechanisms. So our first plan was simply to sequence gynogenic females and assemble the data into a genome, and then assign chromosomes based on differences in female and male coverage and with chemo-based identification. However, something that became apparent quite quickly was that reads from the inversion on the X prime chromosome and reads from the homologous region on the X chromosome were actually collapsing upon assembly, making it impossible to assign scaffolds to the inversion simply because it just didn't exist in the assembly. So because of this, we decided to attempt assigning reads prior to chromosome assembly. And our method of assigning reads to chromosomes utilizes the differences in ploidy between males and females. So both sexes have 2 sets of autosomes. With the X chromosome, males have 1 copy, and then the gynogenic females have either 1 or 2 copies, depending on which portion of the X chromosome you're looking at. And then the inversion is limited to females. So this plot shows a chemo frequency spectrum for male Illumina reads versus female Illumina reads. Male reads on the x-axis and females on the y-axis. And the chemo here is all possible sequences of within a read of length k, in this case 27. And as you can see, 3 quite distinct clusters of chemo frequency emerge. So you have the autosomes, which are 2x in both sexes. The X cluster here, which is 1x in males and either 1 or 2x in females. And then the inversion, which is female limited, so it clusters along the y-axis. And this small cluster here will be mostly errors camus containing read errors. So we use this convenient separation of camas to produce 3 chemo libraries, which we then use to filter reads into 3 individual read files. And simply put, if a read contains a chema from a particular library, then it will be pulled from the raw file. And then we assemble the 3 separate read files separately. And from this assembly, we managed to anchor around 43 megabases to the inversion and around 55 megabases to the X chromosome. And to validate the accuracy of the assembly, what we did was we mapped reads from a gynogenic female and reads from a male against the genome, and then explored differences in coverage between the two alignments. And so this plot shows the difference in normalized coverage between male and female reads across 10 kb windows of the genome. And as you can see, coverage is approximately the same between the two sexes across the autosomes, which is to be expected because they have the same number of autosomes, and also across much of the X chromosome, where again, where ploidy for the two sexes is the same. But where females have an extra chromosome copy, i.e. along the X prime chromosome, which is the inversion and a portion of the X chromosome, coverage is 1x in females and 0x in males. So basically now that the X and X prime are separated in the assembly, it's possible for reads to map where they're supposed to be mapping, and this is what we're seeing. So one of the predictions for evolving sex chromosomes is that they will undergo functional degradation and accumulation of repetitive elements due to lack of recombination. And because the X prime inversion is a female-limited sex-determining region, it's arguably functionally equivalent to a neo-W chromosome. And so we should expect it to follow the same evolutionary patterns of, as other sex chromosomes. However, this isn't what we found for the X prime chromosome. And it seems that the proportion of coding sequence and the proportion of repetitive sequence doesn't differ significantly between the X and X prime chromosomes. And we wonder why this might be the case. And one possibility is that the X prime chromosome isn't that old and hasn't had time to functionally degrade. And in order to test this, we calculated X prime divergence. And we did this using the density of heterozygous sites between the X and X prime chromosome to give us an estimate of DXY. And this gave us an estimate of only 200,000 to 700,000 years. Although perhaps it's worth noting this isn't taking into account the possibility of gene conversion along with other genetic or ecological factors. But anyway, this finding, along with the lack of functional degradation that we found, points to quite a recent split between the X and X prime chromosomes, suggesting that the inversion is probably quite young. And this is actually somewhat surprising, a surprising finding, since monogeny, meaning the production of single-sex broods, along with many of the other unusual genetic traits, appears multiple times throughout the fungus naclade. And as well as monogenic species, there are species where females are digenic, meaning they have normal mixed sex broods, and also mixed species with both monogenic and digenic strains. So it's unclear whether there have been multiple independent origins of these traits, or whether they've been lost there was one origin, but they've been lost in many lineages, and also what role the inversion plays in the evolution of monogeny. And so given this is a family of flies that is thought to have emerged in the Cretaceous period, our young estimate of the inversion is quite surprising. And part of our task is going to be to try and decode the evolutionary history of these strange traits among this family of flies. So as I mentioned earlier, one of our aims is to identify a sex-determining master switch in this species. And one of the useful things about this system is that it's possible to extract embryos of known sex because androgenic and gynogenic females can be identified due to an artificially induced X prime linked wavy wing mutation that you can see in this picture of a gynogenic female. And we can therefore quite easily follow sex-specific gene expression at any stage of development. So recently I've extracted early stage embryos from the two types of females in the hope of capturing maternally deposited transcripts involved in sex determination. And I extracted embryos at 2 time periods in the hope that I can identify these maternal transcripts, which, unlike zygotically expressed genes, should be present at a higher frequency in earlier samples compared to late samples where they're more likely to have degraded. And using this expression data, we've tried to identify genes whose transcription either appears to be broken or genes that are sex-specifically spliced. And we've combined this with annotation data to try and find candidates that might be involved in sex determination through chromosome elimination. And so far, we've identified a handful of genes with roles in sister chromatid cohesion or chromosome modification, whose X and X prime copies might be differentially expressed. And at some point, we hope to perhaps verify these candidates through RNAi or perhaps other knockout techniques. So just to summarize, we've managed to anchor around 43 megabases to the Rhodesia coprophila sex-determining inversion supergene. And we found evidence that the supergene is relatively young and doesn't appear to exhibit functional degradation or accumulation of repetitive elements. And we're working on identifying candidate sex determination genes. So yeah, with that, I'd just like to thank my research group, the Ross Lab at the University of Edinburgh, as well as everyone for listening and also for the opportunity to present my work. So thanks very much.
Everyone, I'm glad today to talk to you about the polymorphism in the sex determination system in wild population of the Nile tilapia. And to start, teleost fish are a wide group with more than 30,000 species and a large diversity of morphs. And interestingly, they also present a diversity of major sex-determining genes, as at least 8 have already been identified for now. And as a comparison, only one gene governs the sex of nearly all of the 6,500 species of mammal. Cichlids in particular exhibit a great variability of sex determination system with several linkage groups, which in our cases will be synonymous to chromosomes, that are endorsing the role of sex chromosome. And as such, 12 different pairs of sex chromosomes have already been identified among cichlids. And both ZW as long as XY sex determining genes can be found on these sex chromosomes. And in some species, the sex can be governed by the interaction of these different loci coexisting, meaning that there can be several pairs of sex chromosomes. And in some haplochromine of the Lake Malawi and Lake Victoria, sex chromosomes may be overridden by the presence of an extra B chromosome carrying W-like sex-determining genes. And these results clearly demonstrate plasticity for the genetic sex determination system in cichlids. And today I'll be talking about the Nile tilapia, which is an African cichlid of economic interest, and thus has been broadly studied, especially through lab strain and commercial stock. And the sex of this species is determined by the interaction of 3 factors. So mainly there's major genetic factors, so following an XXXY pattern, minor genetic factors, also called parental, and the environment can also play a role, and especially if the temperature is high, it tends to masculinize juvenile fish. And today our focus will be held on the major genetic factors, as the pair of sex chromosome is still debated in the literature. And actually, it seems to depend on the strain studied, as some labs have found an association to LG1, even though the sex determination gene, remains to be identified in this region. And whereas some other lab strains were found to have an association to sex on LG23. And more precisely, in the Japanese strain, a tandem duplication of the anti-Mullerian hormone gene has been described on the Y, and one of the 2 copies present various indels, allowing to easily genotype individuals using PCR markers. Still, the AMH genotype on LG23, according to those PCR markers, are not sufficient to explain all the sex ratios observed in a recent study on wild individual of population from Burkina Faso. And these same markers don't work on wild Ethiopian population, suggesting that other genetic factors could interact and that several pairs of sex chromosomes could segregate in the wild. And this led to the main question we had to know whether the diversity we could observe in domesticated strain could reflect polymorphism existing in the wild. And therefore, we decided to work on African wild population from the west, especially from Ghana, and from the east, Ethiopia. And so we first started by studying families using ddRAD sequencing, and we focused on a population called Kapendu, so from Ghana, and the other one called Koka from Ethiopia, you would have understood. And using FST and Fisher exact test between phenotypic males and females, we found a strong association to sex on, in both populations to LG23. And when we dug into this region, we noticed the signal was stronger when getting closer to the AMH region. And the signal was also following an XXXY pattern as expected according to the previous studies on the Japanese strain. So it seems that LG23 carries the sex-determining region in wild population. However, the ddRAD sequencing gives scattered data, and we wanted to focus more precisely on LG23 AMH region. So we switched to a whole genome sequencing method, and this time taking samples directly originating from the wild instead of families, and using the reference genome of the Nile tilapia, which is a female. We were able to compare the reads of Capendu and Coca males and identify the breakpoints of the duplication, which is taking place upstream and downstream of the AMH in OAZ1 and .1L gene, respectively. And we also identified large indels shared by male of both populations that overlap this duplicated region. But we also noticed divergence between the Y of these two populations in males, as for example, some indels, such as the -234 base pair and 5 base pair insertion, were identified in one of the two copies of the AMH gene, but only in Capendu males. And interestingly, these indels have already been described in the Japanese strain. And finally, we noticed various sex-specific SNPs shared between Capendu and Coca males. And using all this information, we were able to create an expected Y chromosome, so composed of a first copy of the OAZ1 gene, followed by a copy of the AMH, then truncated and merged sequences of .1L and OAZ1, a second copy of the AMH, and then a complete copy of .1L gene. And therefore, we aim for however, the use of short reads for analyzing a complex duplication is not optimal, as for example, we could not be sure of which copy of genes were impacted by the indels we found. Therefore, we aim for long read sequencing in order to retrieve the fine structure of this Y region. And we use nanopore sequencing on one male from the two wild population. And what you see here are the results of the long read de novo assembly. And for Capendu, two contigs were reconstructed, whereas for Coca, 3 contigs were built. But in each case, we can find a contig with 2 copies of the AMH genes and the truncated merge sequence of .1L and OAZ1, which are characteristics of the Y chromosome we expect. And so these results confirm the structure of the duplication and allowed us to precisely compare the 2 Ys of this population and place properly the indels we detected using short reads. And as we now knew the expected Y structure, we decided to screen additional Ethiopian population. Surprisingly, the male we chose from the Ora population presented only 1 contig, and this contig corresponded perfectly to an X haplotype. And this led us to wonder if the male we used was an XX male or whether Ora male could lack the duplication of the LG23Y region. And we thus went back to whole genome sequencing again to explore the genome of several males of this population this time, and also several females. And the signal we obtained was quite noisy and confusing, with lots of high peaks bit on all along the genome. But we still decided to have a closer look at the AMH region, even though no proper signal was overlapping this region. And we did confirm what we saw using long reads. So male of this population do not show any sign of duplication around the AMH region. And more than this, we found not a single SNP that would be sex-specific between males and females for the whole region. And this was surprising because Capendu and Coca are geographically far away, so from West and East Africa, and they still share a quite conserved Y chromosome. And as the 2 Ethiopian population are closer geographically, we were expecting them to be also closer genetically and share an even more similar Y chromosome. And thus we proceeded to a phylogeny analysis to check the relationship between these three populations. And we did find, using nuclear phylogeny, that the two Ethiopian populations are closely related compared to the Western African population. And it's the same for mitochondrial phylogeny and for the phylogeny based on the sex region. And these results confirmed that Kapandu and Koka diverged after Ora and Ora and Koka, but still they share the same sex determination system, which suggests that this system is ancestral to the divergence of the Western and Eastern population. So this would mean that Ora males are lacking the ancestral LG23Y sex determining region, and we can now wonder, how is the sex governed in this population? Could it still be genetic and followed, like, for example, polygenic sex determination system? Or could it be a transition and then it's too recent for our method to detect the new SD region? And actually, I cannot really answer, because the method we used would not have really be efficient to detect either of the two possibilities. So we have to dig more into this data using other approaches to try to understand this noise and find something. But maybe it could also be environmental. As I said earlier, the temperature can influence the sex in the Nile tilapia. However, usually masculinization is occurring with temperature above 32 degrees, and it's not a temperature that is known to be encountered in the Ora lake. So in conclusion, LG23 seems to correspond to the ancestral pair of sex chromosome in wild population of the Nile tilapia. But there definitely is polymorphism in this region between wild population, such as indels that are present in only males of the Kapandu population and not in the Koka. And interestingly, these indels that are occurring in the second copy of the AMH create frame shift and then probably lead to a non-functional copy of the AMH. And this is not found in the Koka population. Also, more importantly, the ancestral sex determination system can be lost since Ora population is lacking the LG23Y sex determining region, though we don't yet know what is replacing it if something does. And finally, it's interesting to note that the variability observed in domesticated strain does not truly reflect what happens in the wild, as, for example, we did not find any association to sex on LG1 in any population we studied, on contrary to the domesticated strain. So I'm done, and I would like to thank you all my teammates and collaborators for this. And if you have any question, I'll be glad to answer.
Thanks for the introduction. I'm Djivan Prentout, a PhD student at the LBB in Lyon, in France. And today I will try to answer to the question, do all sex chromosomes exist in plants? So I will briefly start with the Korean view of the plant sex chromosome evolution, and that you probably all know, but it starts with a pair of autosomes on which a Y-specific region and an X-specific region will be formated. And what is important is that we have an arrest of recombination between those two regions. And this arrest of recombination is supposed to expand in the first time. And this expansion of the non-recombining region will have some consequences, mainly on the Y chromosome, which we call the Y degeneration. Indeed, because of the isolation of the Y chromosome in males, we have a reduction of the selective pressure that helps to fight against the accumulation of transposable elements and deleterious mutation. Therefore, this is what we call the Y degeneration, and it should, in the first time, increase the size of the Y chromosome, and we should have a Y chromosome bigger than X, as we can see here. And after a very long time of evolution and long time of degeneration of the Y chromosome, we expect the Y chromosome to become smaller than the X, as we can see here. So the first thing that we can see here is that it seems there is a correlation between the homomorphy of the sex chromosomes and the age of the system. So I define a sex chromosome system as homomorphic if the X and the Y have similar sizes. It not depends on the contents of the sex chromosomes. But what we can see here is that we expect heteromorphic sex chromosome for all systems. But the problem currently with this model is that Among more than 15,000 dioecious species, only about 25 sex chromosome systems have been studied with genomic data, and no studies on all plant sex chromosomes have been done so far. So this step of the sex chromosome evolution in plants is mainly inspired from observation in animals. So here I took a figure from a recent review published this year by Renner and Müller, where we have on the x-axis of both figures the age of the sex chromosome system, and for the figure A we have on the y-axis the number of genes in the sex-determining region, and on the figure B we have the genomic fraction of the sex-determining region. And what we can see is that it seems that so far no plant sex chromosome system that are old and strongly degenerated with large non-combining region have been described so far. So this is why we can ask the question, do old and highly degenerated sex chromosomes exist in plants? So to try to answer to this question, we decided to work with the Cannabaceae family. So the Cannabaceae family is composed of a bit more than 100 species spread in 10 different genera. The family is expected to be around 90 million years old, and there is no strict consensus so far, but the dioecious may be ancestral in the family. And to our study, we decided to focus on two different genera, the Cannabaceae genera and Humulus genera. So those two genera split between 21 and 25 million years ago. And even if there is no strict consensus regarding the ancestral sexual system of the Cannabaceae family, it is well admitted that the last common ancestor of these two genera was dioecious. And cytological analysis showed that Cannabis sativa has homomorphic or nearly homomorphic XY sex chromosome system. Humulus lupulus has an heteromorphic XY sex chromosome system with a Y that is smaller than an X. And XYY sex chromosome system have been highlighted in Humulus japonicus. So what we wanted to do is to study the XY sex chromosome system in Humulus lupulus and Cannabis sativa, and answer to the question, are the sex chromosome homologous and therefore old in Cannabis and Humulus, Cannabis sativa and Humulus lupulus? And if yes, how did they evolve? And there is a second interest that is more applied of working on these two different species. So as you probably know, Cannabis sativa is being legalized in a growing number of countries for its recreational and therapeutic uses. And Humulus lupulus, which is usually called hop, is a beer ingredient because it confers the bitterness and may be useful for the conservation of the beer. But the problem with those two crops is that only the females are useful. In cannabis, only the female produce the THC and the CBD, the two main cannabinoids. And in hop, only the females produce the lupulin, the molecule that is useful for the beer. And the second problem is that there is no sexual dimorphism that is observable before the flowering stage. Therefore, it may be useful to develop technologies to be able to detect or to determine as soon as possible the sex of the plants. And a possibility could be to develop Y-specific markers thanks to the identification of Y-specific sequences. So our strategy to detect the sex-linked genes, we made a segregation analysis. So we need to make a cross, so we cross a male and a female. Then we sequenced the transcriptome of the parents and 5 offsprings of each sex. And then we made the bioinformatic data processing. So it starts with a mapping. What is important here is that we made the mapping of the both species on Cannabis sativa references because the available references of Humulus lupulus were of very poor quality compared to the one of Cannabis sativa. So we mapped both species, both RNA-seq on Cannabis sativa reference. Then we made a genotyping and we used sex detector for the identification of autosomal XY or X hemizygous sequences. Okay, so I will now present the results. So thanks to sex detector, we identified 565 sex-linked genes in Cannabis sativa and 265 in Humulus lupulus. And the first result that was interesting is that among those sex-linked genes, 112 were sex-linked in both species. So after that, we looked at the position of the sex-linked genes on the Cannabis sativa genome. So here you have the chromosomal level assembly of Cannabis sativa genome with the 10 different chromosomes. And in red, you have the density of sex-linked genes found in Humulus lupulus and Cannabis sativa genome, and in blue, the density of sex-linked genes found in Cannabis sativa and Cannabis sativa genome. And what we can see is that the high density of sex-linked genes are concentrated for both species in these parts of the first chromosome of Cannabis sativa. So this leads us to conclude that this chromosome is the sex chromosomes of Cannabis sativa. And also it was another information to think that the sex chromosomes are probably homologous between both species. Then we decided to give an estimation to the age of the both sex chromosome system. To do so, we use molecular clocks and maximal synonymous divergence. And what we can see here is that the maximal synonymous divergence are high and quite similar between both species. And with molecular clocks, we found that the sex chromosome in Cannabis sativa may be around 27 million years old versus 50 million years old for Humulus lupulus. And what is important here is that the differences between our two age estimations is mainly explained because we took a generation time that is in Humulus lupulus that is twice the generation time in Cannabis sativa. So just keep in mind that the synonymous divergence is quite similar between both species, and the both estimations are older than the Cannabis and Humulus divergence. Then to definitely determine if the sex chromosomes are homologous, we made a phylogenetic reconstruction of XY sequences in both species, and we used 4 different outgroup species, as we can see here. And we made the reconstruction with 2 different methods, maximum likelihood 1 and Bayesian 1. And here are the results of the phylogenetic reconstruction of XY genes. So we identify 3 main topologies. The first one on the top is a topology where the X of Cannabis sativa and X of Humulus lupulus are closer between each other than with their Y counterparts. And so this topology indicates that we have an arrest of recombination that is anterior to the speciation. Here we have another topology where the X and the Y of both species are closer between each other than with the X of the Y of the other species. And so this indicates that we have an arrest of recombination that is posterior to the speciation. And here we have a topology that is not really informative with a paraphyletic topology of the sequences in the Humulus lupulus. So I'm not going to speak too much about this one, but the first result that we have with this topology is that we found 11 genes. So this confirms that the sex chromosomes are homologous between both species. And we also can see that genes indicating an arrest of recombination before the speciation are mainly concentrated in these parts of the chromosome, and genes indicating an arrest of recombination posterior to the speciation are mainly concentrated in these parts of the chromosome. Therefore, it seems that there is a null region of the sex chromosome that stop recombining before the speciation between both species, and the youngest region that stop recombining independently in both species. So we also looked at the Y degeneration in the Humulus lupulus and Cannabis sativa, and we found similar patterns of Y degeneration. We identify a Y expression degeneration, which is correlated to the position on the sex chromosome. We also found the dosage compensation in both species. The non-recombining region seems to have similar size, and it represents about 70% of the X chromosome and 9% of the genome of Cannabis sativa. And finally, we estimated the gene loss that is equal to 70% in Cannabis sativa, and that is still undefined in Humulus lupulus because we didn't identify any X homozygous genes, and it's likely because of methodological issues. We could come back to this problem later if you want. So to stop the conclusion, we identified homologous sex chromosomes that are degenerated, that are quite old compared to what have been previously described in plants. Half of the non-recombining region was already non-recombining and last common ancestor of the species, and we have similar patterns of Y degeneration. So this is interesting, but it also raises the question that why are the sex chromosomes homomorphic in Cannabis sativa and heteromorphic in Humulus lupulus? And we have 3 main hypotheses for this. Maybe the Y chromosome of Cannabis sativa is on its way to shrinking and Humulus lupulus evolve faster. Therefore, maybe in few millions generation, the Y chromosome of Cannabis sativa should be smaller than the X. Maybe the rate of transposable elements and genetic loss is very low in Cannabis sativa, and therefore we do not expect an heteromorphy in this sex chromosome system. And the last one is that 2 whole genome duplication happened in Humulus lupulus since the speciation with Cannabis sativa. And maybe just an absence of Y duplication could explain that the Y is smaller than the X, and therefore it's more an expansion of the X chromosome than a reduction of the Y chromosome. So the big conclusions are that we identified homologous sex chromosomes in 2 plant genera, and we showed that it's old and degenerated. We also developed a few markers. I didn't speak about this today, but if you are interested in, we published a preprint in bioRxiv. And there is still some question to determine the exact age of these sex chromosome system. It could be really useful to have X and Y assemblies to have an accurate description of the number of strata, the exact size of the region that was already non-recombining in the last common ancestor. And after, of course, we still have many questions regarding the evolution of sex chromosomes in general. And here was the question of why the sex chromosomes are stable in these 2 genera and are less stable with a high turnover rate in other plant species. Okay, so here are everybody that I would like to thank, mainly Gabriel and Yus, my 2 PhD supervisor. And here are my contact if you want further information. And thanks a lot for the opportunity to present my work and for having listened to me.
First of all, I'd like to thank Wen-Chuan and Michail for organizing this this conference, and I'm really excited to tell you about some work I've been doing at the University of Montpellier on the evolution of sex chromosomes in mouse minutoides. So as Lucas mentioned on the first day of the Congress, there are currently a few hot topics in the field. And from what we've seen since the beginning of the conference, it seems like there are some taxa that are more suited than other to tackle questions that are related to these hot topics. I think that we can all agree that one of the groups that doesn't stand out, at least at first, as being a great taxonomic group to tackle these questions is Therian mammals. They've got really old and extremely stable sex chromosomes. And we tend to say that any chromosomal abnormality is deleterious. And that includes, for certain authors, sex autosome fusions, and especially deviations from the standards XY, XX system. But there are some exceptions. And today, I'm going to tell you a little bit about the African pygmy mouse, Mus minutoides, which is infamous because in natural population, there's a high proportion of sex-reversed females. Now, this is what the karyotype of the African pygmy mouse looks like. And it's characterized by 4 very unusual features for mammals. The first feature is that it has evolved a 3rd sex chromosome that we call the X star. Now, the X star, it derives from the standard X chromosome, and it's analogous to a W chromosome. It reverses the sex of X star Y individuals. And it's responsible for the presence of 1 type of males and 3 types of females in wild populations. So XX, XX star, and X star Y for the females. The second feature, which is not illustrated here, is that the X and the Y are asynaptic in this species, meaning that they don't pair up during meiosis. And that's something you don't see often in mammals. Okay, I've got a bit of a problem here with my slides. I'll just reload it real quick. Hopefully it'll be better. Okay. Third feature is the presence of sex autosome fusions. For the 3 sex chromosomes, the X star, and the Y are all fused to chromosome 1 in most populations of the pygmy mouse, forming 3 neo-sex chromosomes. Now, finally, in some populations of South Africa, the X star chromosome has undergone what we call the whole arm reciprocal translocation. And it was an exchange between chromosome 1 and chromosome 16 from the autosomal pair 13-16. And in these populations, the X star is now fused to chromosome 16 instead of chromosome 1. Wow, that's a lot. Just to recapitulate what we know so far, so in the ancestors of the African pygmy mouse, we had a pretty standard sex determination system, and about 1 million years ago, there was a sex autosome fusion with chromosome pair 1. At about the same time, the X star chromosome emerged, so that's the feminizing chromosome about 1 million years ago. And finally, more recently, so that's probably between 10 and 50,000 years, there was this whole reciprocal translocation, which led to this mess, a lot of sex-linked chromosomes. So obviously we had a lot of questions when we discovered that. And the first question is, how is meiosis achieved? Meiosis is a really crucial process for reproduction. And when you see how many sex chromosomes are linked, for example, in X and Y females, it makes you wonder how it works. And the second question was obviously, how are these sex chromosomes evolving? And I'm talking about the free sex chromosomes and the multiple young neo-sex chromosomes. Now, concerning our first questions, I'm not going to go into too many details. Sorry. We recently published 2 papers analyzing meiosis in fine details, mainly based on cytogenetics. And for the sake of this presentation, I will rapidly tell you about female meiosis. Now, meiosis in XX females is pretty standard. All chromosomes easily find their partner, including the 2 X chromosomes and also the 13-16 pair, which in this genotype is autosomal. Things starts to get complicated in XX star and X star Y females, in which several chromosomes aggregate in this single weird structure during meiosis. So this structure is called the closed quadrivalent, and as you probably guessed, these closed quadrivalents are composed of the neo-sex chromosomes and the sex chromosomes. And here, each chromosome manages to find its homologue in time to ensure proper recombination and balanced segregation of all chromosomes. What's interesting and important to remember for the rest of my talk is that there is non-homologous synapsis that tends to involve chromosome 13 most of the time. So that's the orange one. And that happens in the 2 genotypes. And that chromosome 16, on the other hand, seems to never be involved or less frequently be involved in these non-homologous synapses. And obviously this might point to the fact that recombination might be impaired along this chromosome 13. This leads me to my other question, which is, how are these sex chromosomes evolving? Now, because we're not very original, we did a whole lot of sequencing. We did the whole genome sequencing in assembly of 1 XX female and 1 X*Y female. That was in order to separately assemble the X and the X* chromosome. We did some low coverage resequencing of 14 males and 10 X*Y females that we captured in the wild. And that was to evaluate the recombination patterns between these different sex-linked regions. And finally, we also have data from an RNA-seq study for brain, liver, kidney, for all 4 genotypes, 5 of each, and also males and females from an outgroup, which is Maternally, which has a standard sex determination system. And the objective for this was to characterize the evolutionary trajectories of the sex chromosomes. Now, the first question we tackled is the question of suppressed recombination. And as you can imagine, it's a complex question because there are many different sex-linked regions that could have evolved suppressed recombination. First of all, obviously, there's the X and the X*, but there's also chromosome 1, in which the neo-X and neo-Y could have evolved suppressed recombination. Same for the neo-Y and neo-X*, and finally also the neo-X and neo-X*. And also the 2 neochromosomes, which are composed by the chromosomes 13 and 16. To tackle these questions, we used the assembled genomes and the resequencing data from the XY males and XY females. And we used pretty standard population genomics tools and measures like FST, FIS, and also scanned the whole genome for sex-linked SNPs. So for example, to find non-recombining regions between the X and the X star, we scan for regions of high male versus female FST. And for the neosex chromosomes, sorry about this, for the neosex chromosomes, we searched for regions of elevated FIS, and especially we looked at the sex-limited SNPs, which allowed us to precisely differentiate between the different types of regions that we were analyzing. Now I'll go straight to the results. We first of all found a large region of suppressed recombination between the X and the X star, which passes almost 2/3 of the chromosome. And that's not really surprising because we know there are complex rearrangements between the X and the X star, and also a strong heteromorphy. Now, we also found suppressed recombination between the neo-X and neo-Y in males, a region of about 64 megabase pairs. And the exact same region is also, has also suppressed recombination between the neo-Y and neo-X star. Suppressed recombination was found between the neo-X and neo-X star in XX star females in a smaller region. And finally, we also observed suppressed recombination along chromosome 13. Again, I haven't mentioned that, but it's always in the pericentromeric region. Now, surprisingly, perhaps not, there was no recombination suppression along chromosome 16. And what we think is happening here relates to what I told you earlier concerning the fact that chromosome 13 engages in non-homologous synapses, but not chromosome 16. Okay, time to take a break. I'm gonna recapitulate what we know based on this data. And this system is really interesting because it's characterized by different things. First of all, it's characterized by various recombination patterns. Now, especially what's interesting is that we have large non-recombining regions along the X star and also along the neo-Y. Second of all, we have various transmission patterns. What's interesting is that the X star has acquired a region that is restricted to female lineage, and that the Y has lost its male limited transmission. And finally, we've got these different regions of variable ancestry. So some regions used to be sex chromosomes, some regions used to be autosomes. And obviously, this led us to the question, how do these parameters influence the evolution of the sex-linked regions? And we could make a few predictions based on the canonical model of sex chromosome evolution. The two predictions I'm going to tell you about is we could potentially see degeneration along the non-recombining regions of the X star / neo-X star / neo-Y. And the other thing is that we might expect to see sexualization of the gene content on all of these regions, and especially an enrichment in genes with sex-biased expression. Concerning the evolution of the sequence, as I told you, these sex chromosomes are really young, so perhaps it's not too surprising that not much is going on in terms of sequence evolution. I'm not going to show any details here, but there's very low divergence in all of the sex-linked regions. We haven't been able to find any accumulation of repeated elements or repeated sequences, no large deletions, no substantial gene loss or impairment of gene functions, etc., etc. That's like the standard things people look at when they want to study the evolution of sex-linked regions. However, we were not completely out of hope because studies on other young sex-determining systems have shown that changing in the gene expression can arise before any substantial sequence evolution can be seen. So we looked at the evolution of gene expression. And as always, it's a bit complicated with this system because the search for sex-biased genes is more challenging in this species than in standard sex determination systems. The emergence of the X star has generated new axes of variation and selection, and in addition to the standard male-biased and female biased genes, we might also encounter genes that are differentially expressed in relation to genotypic sex. So that's the dichotomy between sex-biased genes that can relate to phenotypic sex and sex-biased genes that can relate to genotypic sex. And for example, there could be genes that are more highly expressed in individuals that carry the Y chromosome than individuals that don't carry the Y chromosome. So to disentangle all of these effects, we did all possible 2x2 comparisons between the 4 genotypes. And we separated the unbiased genes. So these are the genes in which all 4 genotypes show similar expression from the genes that are biased in at least one contrast. These genes, we ran them through a program called WGCNA, and it basically clusters genes based on their expression patterns in the 4 genotypes. These different clusters, we were able to correlate them to different traits. For example, we have a large cluster of female biased genes, which in all females have the same expression, and male have a lower expression. So it's this one, sorry, female biased genes. And these clusters can be separated in 2 groups, those that relate to phenotypic sex and those that relate to genotypic sex. First thing we did with these clusters is to see whether there's any enrichment in these genes on the sex and the neo-sex chromosomes. So this plot, what it displays is the proportion of genes in each cluster that are carried by the different genomic compartments. So the autosomes in gray, the X chromosome / X star light blue, and the neo-sex chromosome in the darker blue, and then also a few genes on chromosome 13. And as you can see, there's no apparent enrichment in female biased genes, nor male biased genes on the sex and neo-sex chromosomes in comparison to the unbiased genes. However, there's a huge enrichment in the other 3 clusters that we've analyzed. And what this suggests is that we've got an enrichment of sex-linked genes, especially, or I should say only the genotypic sex-linked genes. And that's especially true for the neo-sex chromosome, which strongly suggests sexualization of the neo-sex chromosomes. I see that I'm running out of time. I'm going to go really quickly on my last slides. I'll skip straight to the conclusion. I'll just tell you that we focused our attention on 2 clusters, because these clusters were able to tell us more about the degeneration of the neo-Y and the generation of the X star chromosome. And what we were able to show by comparing the results to the sister species of most minutoides is that our data shows a downregulation of neo-Y linked genes, which is concordant with subtle degeneration of that chromosome. And on the other hand, we didn't see any downregulation of X star linked alleles, which suggests that the X star is not degenerating. Okay, so to conclude, I would just like to remind you that we've got a very complex sex-determining system in this species with multiple sex-linked regions, which have different segregation patterns and different mechanisms of recombination suppression. Concerning the evolution of these regions, we show an enrichment in sex-biased genes, especially on the neo-sex chromosomes, and not so much on the ancestral sex chromosomes. And we also see no signs of degenerations along the X star, but subtle signs of degeneration along the neo-Y. And this obviously leads to a whole load of new questions. Perhaps these observations are just linked to the fact that there are different times of recombination suppression. And the other possibility, which I think is more interesting, is that these 2 regions, so neo-Y and X star, are under different evolutionary constraints related to their potentially ancestry. The X and the X star have been sex-linked for over 150 million years, and the X star in mammals has highly specialized gene contents. It's possible that it's under strong stabilizing selection, which limits its adaptive potential. Also, the X star is hemizygous in X star Y females, which should shelter it from the accumulation of deleterious mutations. On the other hand, the neosex chromosomes are like kind of a blank canvas for sexual selection. So perhaps it's not surprising that they have accumulated more new sex-biased genes than the ancestral sex chromosomes. That's it for me. I want to thank all the people involved, especially people at the University of Montpellier, but also people from the UNLAM Institutes in Norwich, the EGH in Montpellier, and our colleagues in Madrid. And that's it for me.
So again, thanks to Wenjuan and Michail for inviting me to give a talk here today. I want to just point out right off the top here that everything I'm going to discuss is work from a PhD student in my lab, Shannon Keating, and she is kind of our rad seat guru now, and she is doing lots of cool stuff, and this is one of the latest things that she's done. So I think everyone here is familiar with the diversity of sex-determining systems that we find across the tree of life, and you know, just focusing in on vertebrates here, you can see these different colored squares indicate the different sex chromosome types or environmental sex determination, presence, absence across these different vertebrate clades. And the first thing you notice here, as well as looking in other parts of plants and animals, is that the diversity of sex chromosome systems is not distributed evenly across the phylogeny. So there are some clades that have very few transitions, things like mammals, crocodilians, and birds. And there are other clades that have lots of transitions. You know, so again, in this tree, you can see fish, amphibians, and reptiles really have a lot of diverse sex-determining systems. And what's important to keep in mind here is that at least amongst vertebrates, the majority of sex chromosome transitions remain unidentified. So when you start digging into fish, amphibians, and reptiles, there's still more we don't know than we really do. And even when you look at kind of a finer scale, so if we're going to look here at just, let me get my laser pointer here, just the lizards and snakes, you see a similar pattern kind of at this finer scale. So here's a phylogeny of the lizard and snake families, and you see, again, lots of diversity. The other thing I want to point out here is these families that are in white are families where we lack information, and there's still a surprisingly large number of those across the lizard and snake tree. But even here, we see this same pattern where you have some really stable groups and other groups that have lots of turnover. So in terms of stable groups, we have the canophidian snakes, we have the pleurodonts, things like spiny lizards, iguanas, and anoles, laceridids, skinks, etc. So these are large species-rich groups that all, for the most part, have just a single sex chromosome system. And similarly, we see clades that seem to have lots of transitions. And I've highlighted those clades in these gray boxes here. So here the hanophidian snakes, the boas and pythons, the acrodonts, these are dragon lizards and chameleons, and then the geckos here. So again, the diversity of sex determining mechanisms is high, but that diversity is not evenly distributed across the tree. And those turnovers certainly aren't evenly distributed across the tree. So we're going to focus here on geckos today. And here's a phylogeny of the 7 gecko families, and we're going to highlight this one right here. This is the Eublephairidae. They're sometimes called eyelid geckos because they're the only group of geckos that retain eyelids. The majority of geckos species actually lack eyelids. And this is a phylogeny of the 6 genera. So we know a little bit about the sex determining systems that are going on here. So we know Eublepharis and Hemithiconyx have temperature dependent sex determination, but Alluroscalabodes and Goniurosaurus are completely unknown. And we know that Coleonyx has an XY sex chromosome system, and I'll talk more about that obviously in a minute. The other really nice thing about using Eublephairidae as a model here in this example is that they have one of the few gecko genomes available. So again, draft gecko genome was published a few years ago, and we're going to use this a little bit later here in this talk. So I just want to point that out, and that is this genus here. So Eublephaриdae live kind of throughout the tropics globally. Coleonyx live in the Western Hemisphere here, and from the southern United States into Mexico and south into Central America. And there are, while there are 8 valid species, there are really just 4 that are widely available to researchers. The others are hard to collect and pretty uncommon even in the wild. So from here on out, we're just going to talk about these 4 species. So what's really nice is that these 4 species are kept in the pet trade, and there are incubation experiments that have shown that all 4 of these have genotypic sex determination. And this is based on incubation experiments. So you incubate eggs at different temperatures, and for all 4 of these Coleonyx species, you get even sex ratios at all those incubation temperatures. Kind of moving forward from that bit of information, back in 2010, Picorna et al. Published a cytogenetic study of Coleonyx elegans, showing that they had XY sex chromosome system and actually a Y fusion here. So you have 4 X's in females, and then here's your fused Y, and then the 2 X's there in a male. And then just last year, Lucas and Michail's group published a really nice paper where they dug a little deeper into the sex chromosomes of Coleonyx elegans. So they looked at read differences between males and females to identify X-linked genes. And then they did qPCR of genomic DNA in both males and females to look for conservation of that sex chromosome system in other eublepharid geckos. And so what they found is that in Coleonyx elegans and mitratus, you have the same sex chromosome system, and this is syntenic with chickens' chromosome 1, 6, and 11. But you don't see that in other eublepharids, particularly in the other 2 Coleonyx that are available to researchers, Coleonyx brevis and variegatus. So that brought us to where we are here today, and we had access to a bunch of Coleonyx brevis material and decided to do our RAD-seq on that to see if we could identify what was going on in Coleonyx brevis. So this is the species here. They're completely adorable. So we went ahead and did RAD-seq on 8 males and 9 females. We found 576 male-specific markers and no female-specific markers. So this enrichment of male-specific markers indicates an XY sex chromosome system. We did PCR validation of 4 of those, and you can see they amplify broadly in a male-specific manner here. So we can confirm that they actually do have male heterogameity. The next thing we did, because we have a draft genome of a closely related species with Eublepharis, is we used BLAST to query those male-specific RAD markers to Eublepharis genome. And then we took those scaffolds that got hits and then blasted that to the chicken genome to identify the chicken chromosomes. So we had 42 of those male-specific RAD markers have just a single hit in Eublepharis. And then most of those correspond to chicken chromosomes 2, 4q, and 28. So, and these are different than the chicken chromosomes that were identified in Coleonyx elegans and mitratus. So if we kind of come back to this here, so we know that mitratus and elegans share this XY sex chromosome system using chicken chromosomes 1, 6, and 11. So that likely evolved here in this common ancestor to mitratus and elegans. We know that brevis has this XY system using chickens chromosome 2, 4q, and 28. We actually tried to amplify some of these male-specific primers that work in C. brevis in Coleonyx variegatus, and we didn't get any amplification. Although that doesn't mean that they don't share the same sex chromosome system. It simply means these primers don't amplify in a sex-specific manner. So as of now, what's going on in Coleonyx variegatus remains unknown. So whether this XY system evolved along this branch in Brevis or in the common ancestor of Brevis variegatus remains open right now. Nonetheless, there is a transition in sex chromosomes within this genus. So the Brevis sex chromosomes are still XY, Mitratus and elegans are still XY, but there's a change in the chromosome or linkage group that they're using as their sex chromosomes. And this, I think, highlights, again, the diversity that you see in sex chromosomes and the number of transitions that you find in some of these clades that are enriched for sex chromosome transitions, things like boas and pythons, dragons and chameleons, as well as geckos. And finding transitions within a genus is still relatively uncommon, at least amongst amniotes. Fish people and frog people, I know you think this is like happens everywhere, but in amniotes at least, this is still relatively uncommon. However, in geckos, we've identified at least 5 gecko genera that have intrageneric transitions. And so the genus gecko, Hemidactylus, Cerdidactylus, Brendan Pinto, a former PhD student in the lab, just put up a preprint on bioRxiv showing a transition in Spherodactylus, and then the story I'm telling you today about Coleonyx. And this is without really looking at a lot of geckos. There are close to 2,000 described gecko species, and there are a lot of geckos that remain unstudied. And I think this is a situation where in at least several of these families, the more you look, the more you're going to find. So with that, I'd like to thank NSF for funding. I'd like to thank Carl Lieb for facilitating collecting permits in Texas, Stu Nielsen for some of the photos, and then some of the undergrads that work here at Marquette doing animal care and maintenance. And with that, I think I've got plenty of time for questions here.
So I'm going to be talking about some work from my group done primarily by a former graduate student, Kiran Adhikari, another former graduate student, Jayhawk Sun, and a former postdoc, Pablo Delclos. I'm actually going to be talking a lot about phenotypes today, phenotypes affected by sex chromosomes and how they can contribute to the maintenance of a polymorphism and how they interact with the genotype. This work was done by those graduate students and postdoc, as well as a bunch of undergrads in the lab and in collaboration with colleagues at University of Zurich and University of Groningen. So just kind of, I know we're all familiar with this, but orient us. What I'm talking about here is sex chromosome turnovers in which we have, say, an ancestral karyotype with a male determining gene on a Y chromosome. And so you have XY males and XX females. We can get evolutionary transition or turnover such that we have a derived situation with a new male determining gene. We'll call that M2, arising on a proto-Y chromosome, which was an autosome. The old sex chromosomes revert back to autosomes. This is thought to happen because a male beneficial allele is genetically linked to the male determiner, driving the invasion and fixation of that new sex chromosome or new Y chromosome. But in order for this to happen, we need to have a period of what we call polygenic sex determination, in which both the ancestral and derived karyotypes segregate as polymorphisms in a population. And so my lab is interested in understanding this polygenic sex determination process. And specifically what perplexes us is that a lot of population genetics theory predicts that this will be an unstable intermediate between monogenic equilibrium or equilibria, and we're interested in understanding, can this actually be maintained as a polymorphism? So the model system we work with to examine this is a housefly. The housefly has a male determining gene called MDMD, or abbreviated M. It's a negative regulator of this gene transformer. So if you have the male determiner, transformer is off, you develop as a male. Without the male determiner, transformer is on, you develop as a female. The male determiner can be found on what's called the Y chromosome, so you get XY males and XX females. But the reason we study it is that the male determiner can be found on other chromosomes as well. And it's commonly found on the third chromosome. We call that 3M. And we've, in a series of papers, have shown that these YM and 3M are both young proto-Y chromosomes. So they're minimally differentiated from their homologs X and the standard third chromosome. In addition, you can have males that carry multiple male determining chromosomes in their genome, so YM and 3M, or 2 copies of 3M, or all sorts of other arrangements. And then on top of all of that, there's a second allele of transformer called TRA-D. It's a dominant allele. It's desensitized to the male determiner. And so if you have the TRA-D allele and you have male determining chromosomes in your genome, you still develop as a female. So that 4th chromosome that carries TRA-D is actually a W chromosome. So we have all this crazy stuff going on. This system has been stable in populations for as long as it's been studied. Frequencies don't really change from decade to decade. And so we're interested in understanding what maintains this polymorphism. And I'm going to focus today on the YM3M polymorphism. So getting to sort of the phenotypes that I mentioned at the beginning, people have looked for phenotypic effects of these chromosomes to understand what maintains the polymorphism. And so one way people have done this is Rhonda Hamm conducted experiments where she combined a YM male and a 3M male with an individual female in a container in the lab and asked who mates. And if you expected there wasn't any effect on the chromosomes in terms of mating success, you get half the time YM successfully mates, half the time 3M. She's found that actually 3M disproportionately is the successful male in these competition experiments. My lab has done similar experiments where we've taken 5 males and 5 females. All the males are the same, so this is effectively a no choice experiment. We ask within a 4-hour period how many males mate, and we see that more 3M males mate on average than YM males, and so we think that the 3M chromosome confers a mating advantage through faster mating success. Rhonda also did experiments with population cages. She started them with 50% YM, 50% 3M males, and tracked the frequency of the 2 chromosomes over generations. And she found that in 4 replicate populations that she set up, all the time, the 3M chromosome eventually reached what we call near fixation, or a very high frequency, and YM was effectively lost or close to lost. So now we have this problem. 3M seems to be much better than YM. Why does this polymorphism get maintained? And so we turn to the distribution of these chromosomes in natural populations to try to get at that question. And so we know from a lot of work, including work by Rhonda, that YM is common in the north and 3M is common in the south. And this is observed, I'm showing you here from the eastern United States. We see this in Europe, we see this in Asia and Japan. This is a common pattern. And so we hypothesized then that, or we observed also that it's cold in the north and it's hot in the south. I'm not a trained ecologist, but at least I was aware of this phenomenon. And so I hypothesized that 3M confirms a fitness benefit at warm temperatures and YM confers a fitness benefit at cold temperatures, such that you get this G by E, genotype by environment, or specifically we call genotype by temperature, G by T interaction. So that's our hypothesis. We tested that hypothesis by putting the YM and 3M chromosomes on a common genetic background, as we refer to these as nearly isogenic strains. And then we measured various phenotypes. And so Kieran looked at heat tolerance. So he stuck the house flies at 53 degrees Celsius, which is insanely hot, but it's what we needed to do to get a phenotypic response. And he asked how long can they survive? And he raised the flies at 2 different temperatures because flies are ectotherms, and ectotherms actually, their thermal traits depend on their developmental temperature. And so we expect then that the flies with that southern 3M chromosome raised at a hotter temperature would have the most heat tolerance. So this is the results of his experiment. The y-axis here is showing you heat tolerance. So higher values indicate longer survival at 53 degrees Celsius. We have an acclimation effect, like I told you. So you develop at 29, you're more heat tolerant than at 18. And we have a genotype effect. So the 3M chromosome confers heat tolerance or more heat tolerance. So that's nice. That's consistent with the distribution. Kieran also looked at cold tolerance. He stuck the flies in a refrigerator at 4 degrees Celsius. and he asked, do the flies with that northern YM chromosome who develop at a colder temperature have better cold tolerance? So now the y-axis is showing you cold tolerance, and there's the acclimation effect. You develop at a colder temperature, you do indeed have more cold tolerance. And if you have that YM chromosome, you are the most cold tolerant. So once again, consistent with the natural distribution. So YM confers cold tolerance, 3M confers heat tolerance. But flies, as the name suggests, can fly. So if it's too hot or too cold, they will move away and find a temperature that they prefer. So what Pablo did is he constructed this apparatus in the lab where he could measure thermal preference. So it's just a sheet of metal, and because one end is in a heat bath and one is in an ice bath, you get a thermal gradient across the metal, and then you can ask, where do flies like to be along that thermal gradient? And that's a measure of their temperature preference. So now we have the y-axis showing thermal preference, and the x-axis is again showing you the developmental temperature. Each point here is an individual fly that Pablo measured that had a particular genotype and developed at a particular temperature. And we have an acclimation effect for thermal preference as well. In this case, if you develop at a warmer temperature, you prefer a cooler temperature. But I want to draw your attention here to the genotype effect, which occurs at our intermediate developmental temperature. Once again, we see that the 3M males are warm adapted, so they prefer a warmer temperature. The YM prefers a cooler temperature, consistent again with the distribution. So we have now this consistent signal that YM confers fitness benefits for cold and 3M for hot. And so we think that this maintains the polymorphism across the populations. So returning back to those experiments I showed you at the beginning, we wondered, is this 3M advantage because these experiments were performed at a warm temperature? And can we get a YM advantage at a cooler temperature if we do these same experiments? So what Pablo did is he said, well, let's take our no choice assay and do it at 2 different temperatures. So now it's being done at 22 and 29. And what we see is that the 22°C males are more likely to mate within the 4-hour period. So they outperform. So colder temperature leads to a better mating performance in these flies. And he said, is there a genotype effect? Well, yes, there is a genotype effect, but it's independent of temperature. So it doesn't matter if you're looking at 22 or 29°C, the 3M males do better. So we would need to have an interaction term in our statistical model in order for there to be a G/E effect. And there's no interaction term that's significant. And so once again, we have this problem where when we try to look at these fitness related traits like mating, we see that 3M males win all the time. So what Kieran did is he said, well, maybe we can detect a fitness difference in a population cage conducted at multiple different temperatures. So what he did is he set up population cages just like Rhonda Ham had done in the data I showed you before, where they were half 3M, half YM, and he tracked their frequency over generations. And so 100% on this y-axis indicates that the 3M chromosome reaches fixation, 0% means it's lost, and which means YM reaches fixation. And so this is the result at 18°C. And I'll come back to this female coloring in a second, but I just want to draw your attention to what happens after a few generations, which is in 3 of his 4 replicate populations, the 3M chromosome reaches high frequency even at 18°C. So this should be our warm adaptive chromosome, and our warm adapted chromosome does well at a cold temperature. He did this same experiment at 29 degrees. It goes for more generations because development's faster at 29, so you can do more generations. And what's crazy here is that the YM chromosome reaches high frequency at 29 degrees, and that should be our cold adaptive chromosome, and it's doing really well at warm temperatures. And so now we were like, what's happening with these fitness differences we expect across temperatures? What's going on? And we looked specifically at the first generation. And what we see is that what the colors on the graph indicate is where do the females come from in these population cages? And so half of Kieran's cages were started with females from a 3M strain, and half of them were started with females from a YM strain. And so at 18°C, when the females come from a YM strain, the 3M chromosome increases in frequency dramatically in that first generation. And so we think what's going on is there's an unfamiliar male advantage such that the 3M males do better at mating with females from a YM strain, but only at 18 degrees Celsius. And on the opposite side of that, we see that the YM chromosome seems to confer an unfamiliar male advantage for those males at 29 degrees Celsius. So when the females come from a 3M strain, the YM males do really well. And we think this has to do with mating success because housefly females rarely mate a second time. And so any changes you see within a single generation probably has to do with the mating success of individual males. So we think we might have temperature-dependent negative assortative mating going on as well, which we're trying to test with some follow-up experiments. So I've shown you then in houseflies that a lot of important phenotypes seem to depend on genotype and temperature. And so I want to sort of generalize here and say that I think we should think about ecology when we think about the evolution of sex chromosomes. And this is not a novel idea. People have been doing this in other systems. For example, we know that in lizards, sometimes a chromosomal sex determination system can be overridden by temperature, as we've been hearing about in some of the presentations or in the symposium. And we've also heard about guppy Y chromosomes that seem to affect male traits, but also affect vulnerability to predators and predation, of course, being an ecologically relevant phenotype. And W chromosomes in cichlids can be linked to camouflage alleles, as in the case demonstrated here. And so that you have basically sex-linked camouflage, and once again, camouflage hiding you from predators. And so I sort of want to step back and generalize and say, we should continue to think about the ecological effects of sex chromosomes as a way of potentially understanding the unique evolutionary dynamics of these sex chromosomes that we're all studying here. So thank you very much for your time. And I appreciate the organizers of the symposium for putting together a great series of talks. Thank you.
So amphioxus, the deeper branching chordates, is closely related with vertebrate. And there are about 30 species in this group. And their karyotypes seem to be conserved. The deeper number range from 36 to 40. Last year, one group reported the ZW sex chromosomes in Florida amphioxus. So if this is true, then the sex chromosome must be homomorphic because we do not detect differentiated sex chromosomes. So then it's important we assemble the fixed diploid genomes because the Z and W chromosome could be very similar, like the autosomes. So here we hybridize the Florida amphioxus with Japanese amphioxus and also with the Chinese amphioxus. Then we sequence the hybrids. So in the Japanese Florida hybrids, we have the Z chromosomes. And in the Chinese Florida amphioxus hybrids, we have the W chromosomes. So then we can assemble the Z and W chromosomes separately. Then using resequencing data of males and females, we can map the sex-linked regions. And it's clear there are sex chromosomes, and they are all ZW because those variants are all heterozygous in females and homozygous in males. And those sex-linked regions are different in terms of their genomic location, and they also have different size. In Florida amphioxus, here in blue regions, this shows the largest divergence between the males and the females. And its boundary also agree with the breaking point of ZW, the inversion between Z and W chromosomes. So this blue region must be the oldest strata. Then the set determined genes must be also in these regions. but we do not see any usual suspect of sex-determining genes in these regions. In Japanese emphasis, the sex-linked region is smaller, but again, we do not see any usual suspect of sex-determining genes in the sex-linked regions. In Chinese emphasis, all the sex-linked variants are all within a single genes. So this becomes the only candidate sex-determining genes. But we do not know what this gene really is because there's no homologous in vertebrates and had not been studied before. But we do see testis bias expression. So this agree with its role of sex determination in this species. Then we further explore whether vertebrate conserved sex determining genes are present in amphioxus genomes. So for the 10 genes we checked, 3 are absent in amphioxus, including AMH and the DMRT1. So other DMRT families are present in amphioxus, but not for DMRT1. So in summary, we show a large diversity of sex chromosomes in amphioxus, and the sex-determining pathway is probably different from what we know in vertebrates. Thank you for listening.